morning, students. Yesterday's class we have seen the useful role of bacteria in the case of medicine and especially about antibiotic we have seen. Today's class we are discussing about serum, how it is produced and how it is used in medicines. So let us see what is meant by serum. So serum is a blood plasma from which fibrinogen has removed. So serum is a blood plasma. What are the components of blood? Yes, that is RBC, WBC, platelets and plasma. So here we are taking only plasma and from the plasma we are removing what? Fibrinogen. And after removing this fibrinogen you can see certain antibodies is present in this one. These chemical substances are including antibodies and antitoxin. And now how this serum is produced? This plasma we have taken and removing the fibrinogen, and then it is ready to produce these antibodies, and you can see that it is used as a preventive measure against certain bacterial attacks. So this blood plasma, after removing the fibrinogen, it can be used to prevent certain attacks or invasion of this bacteria. And what we are doing, it is introduced in the blood of a healthy animal. Say for example, a cow or a horse, we are introducing this one, and afterwards you can see that the body produces antitoxin. So a chemical which can fight against this toxin. So our body will produce an antitoxin, and after injecting several times this into the body, you can see this, the horse body or cow's body, it will produce this antitoxin, and that will be taken out, the blood will be taken out, and only plasma we are removing, and then the clots also we are separating, and the remaining substance we are calling it as serum. And it is, you can see that usually for snake bite and all. The same thing we are using, that anti-venom. Usually when a person is taken to the hospital after snake bite, the doctors usually they will ask what kind of snake is bitten by to this person. So you can say that then they can use the correct anti-venom. The same method only, that small doses of the particular snake's venom is injected into the body of this animal, I mean cow or horse. So very small doses only. After injecting it for several times, you can see that this from the animal's body the blood is taken out, and we are separating the plasma. So this is used mainly used in the treatment of anti-venom for snake bite and all. And again in the genetic engineering, you can see that made it possible in, it is introduced, the human genes are introducing into this, and we can see certain bacteria like Escherichia coli, and this one we are using in the case of the treatment of diabetics, that is insulin we can produce by this method. And a few serum compounds, you can say that it is used in the blood clotting factor 8 for hemophilia type of A, and blood clotting in the case of hemophilia B type. So we can see that both cases we are using this serum. Next we can go to the vaccines. So antibiotics over, serum is over, next one is vaccine. So you may have heard about many vaccines. Can you say some example of vaccine? Yes, you have heard about against tuberculosis we are taking vaccine, against typhoid we are taking vaccine, against then whooping cough, like that many diseases we are taking vaccine. Polio you are seeing that in the orally we are giving as a drops. So these are some examples. And what is meant by vaccine actually? It is a killed or weakened germs. germs of a particular disease. And the process of injecting this or introducing this into the body is called as what? Vaccination. Vaccine is a preparation consisting of weakened germs or dead germ diseases. So, weakened or dead germ diseases, it is that we are introducing these germs into a healthy person's body. Then that process of introducing this vaccine into a healthy person's body, that is called as what? Vaccination. Here you can see that in the medical field there is a variety of vaccines are available. Now you might have heard about the co-vaccine for corona. It is getting ready. So actually what is doing? Yes, the weakened or killed. Two method is there. Either we are taking weakened or next we are taking this killed germs and introducing into a healthy. First they will introduce into a healthy animal's body, as in the case I have told you that it may be in the case of a horse or a cow, a rat or a monkey, like that. Any animal's body it is introducing so that the body will produce certain antibodies. So the resisting capacity the body will get, and so that after doing this for several times, you can see that vaccination will be ready, and the body will be produce an during vaccination. You can see that the body will produce an antitoxin, a substance produced in the animal's bodies which react with the persons and produced by the invading germs. And what are the two very common type of vaccine? One is called as TAP and other one is called as this one BCG. You are familiar with BCG. All of you got BCG vaccination in your younger age itself, as soon as you are born. So you can see this TAP vaccine, that is against typhoid. T typhoid, A paratyphoid A, and B paratyphoid B. So this is called as TAP vaccination against typhoid, and next is BCG vaccination, that is against tuberculosis. And what is the full form of this BCG? Bacillus calmette-guerin. That is the full form of this BCG. Next, let us see that in medicine what is meant by toxoids. So, what is meant by toxoids? Toxoids are inactivated toxins of the particular bacteria which can still stimulate the production of the respective antibodies, such as those for producing immunity against diphtheria and tetanus. So, these are also inactivated. Toxins are toxoids are inactivated toxins. Toxin means poisonous substance. Now let us see the bacteria. What is its role in agriculture? It has a vast role in agriculture. First, let us see the nitrogen fixing bacteria. So, first let us see the nitrogen fixing bacteria. A special category of bacteria which is called as what? Rhizobium. These lives in the root nodules of certain plants, especially leguminous plants. Can you say some examples of leguminous plants? Yes, all the types of bean plant, it comes under the category of leguminous plants. And if you pull a leguminous plant, say for example a bean plant, you can see its roots. Generally you uproot a plant, you can see that roots containing, suppose this is the roots, it contains taproot system as you know already, and the roots branches will be like this, and here and there in the roots a bulky portion you can see like this. These are called as root nodules. A textbook plant also is a leguminous plant, generally without pricking the thorn, generally you pull out or uproot a touch-me-not plant if beans plant is not available. You can see such structures here, and these are called as root nodules. And what is lodging there? Yes, a type of bacteria which is called as Rhizobia Bacteria is living in this root nodules. And what is the speciality of this Rhizobia Bacteria? Yes, it can trap the atmospheric nitrogen. So usually all plants for its synthesis, or you can see that it needs what? It needs this nitrogen. But plants do not have the capacity to trap this atmospheric nitrogen, though 78% of nitrogen is present. 78% of the nitrogen is present in the atmosphere. No plants can trap this nitrogen directly. So what happens? Only in the form of nitrates, plants can take this nitrogen or absorb nitrogen, that is from the soil. So this Rhizobium bacteria, you can say, which is lodging in the root nodules of Rhizobium plant, it can absorb or it can consume this free nitrogen from the atmosphere. So this free nitrogen will be trapped by this Rhizobium bacteria, and it can convert this nitrogen into nitrate, the soluble form, that is nitrates. Then these nitrates can be easily absorbed by this pea or bean plant as well as other plants. This nitrate, if it is in the soluble form in the soil, that can be absorbed by the pea plant or bean plant or any other plant easily. So that is called as nitrogen fixation bacteria. Example is Rhizobium. Next you can see that certain bacteria in the soil, we can see just like Azotobacter, Clostridium. These are the other two examples of bacteria. These use free nitrogen in the soil and convert it into ammonium compounds. So you can see that ammonia combines with the hydrogen and forming these ammonia compounds, and then this amino acid and nitrates, and thus it will enrich the soil. So Clostridium bacteria as well as Azotobacter. These are the two types of another nitrogen fixing bacteria. These bacteria live in the soil, and this you can see that they also can take this free nitrogen from the atmosphere and convert it into ammonia and hydrogen, and then it is converted into amino acid and nitrogen, and again you can see that there can be nitrates that can be used by the plants which is present in the soil. Next, coming to the nitrifying bacteria, a group of called as nitrifying bacteria, another group is called as denitrifying bacteria. Now let us go through what is meant by nitrifying bacteria. As you know, for the synthesis of proteins, plants need this nitrogen. I told you, nearly in the atmosphere 78% of nitrogen is present. Even then, plants cannot consume it directly. Only in the form of soluble form, that is in the nitrate form only, it can use it. So you can see that they make use of this free atmospheric nitrogen that can obtain only in the form of nitrates. And you can see certain type of bacteria, they will convert it into the form of nitrates, and this process is called as nitrification. Fixing the nitrogen in the soil by certain bacteria is called as what? Nitrification. Fixing of nitrogen in the soil in the form of nitrate by certain type of bacteria is called as what? Nitrification. So they want this bacteria, first they want to convert this nitrogenous waste of animals nitrates and then it is absorbed by the plants. So here you can see nitrification is the process of fixing atmospheric nitrogen in the soil. Once more, nitrification is the process of fixing nitrogen in the soil. Free gas nitrogen is in the atmosphere that first want to fix in the soil. By again you can see that the dead plants and animals body, when it decay in the soil, you can see it release a compound called as ammonia. This ammonia is then converted into ammonium compounds, and this ammonium compounds first converting into this nitrites by a certain group of bacteria, and this is called as Nitrifying. So this bacteria is called as Nitrifying, and again this bacteria Nitrifying, nitrite is present. Another bacteria which is called as Nitrobacter converting this nitrites into nitrates, and thus the nitrogen fixation takes place. And what is the role of denitrifying bacteria? So you have seen nitrifying bacteria, two you have seen, one is converting this ammonium compound into nitrites, that is Nitrifying, and then this nitrites are converting into nitrates, that is Nitrobacter. Now denitrifying bacteria, a group of bacteria that what they will do, these bacteria are present in the soil, they break down the soil nitrates which is present in the soil and then release the nitrogen gas into the atmosphere. So these bacteria, these denitrifying bacteria are a group of bacteria which is present in the soil, and these bacteria what they will do, they will, you can see that they will break down these nitrate compounds which is present in the soil and releasing free nitrogen in the atmosphere. So one such example of denitrifying bacteria is Pseudomonas. So this is an example of denitrifying bacteria.